The overall goal of this procedure is to investigate neurotransmitter vesicle dynamics in neuroblastoma cells using pH-sensitive probes and total internal reflection fluorescence microscopy, or TIRF. This is accomplished by first plating cells onto glass cover slips and transfecting them with genetically encoded pH-sensitive fluorescent probes of vesicle fusion and recycling, which are selectively targeted to synaptic vesicles. Protein expression is expected within 24 to 48 hours. The second step is to record vesicle dynamics by total internal reflection microscopy. This step is particularly critical for the success of the experiment, and a step-by-step description on how to achieve TIRF configuration is provided. Next, once the correct TIRF configuration is reached, the sequential images can be automatically recorded by the software in basal conditions or stimulated conditions. The final step is to process images and extract data from the videos using commercial software or homemade algorithms. Ultimately, using TIRF microscopy to record the changes in the fluorescent signal deriving from vesicle fusion to the plasma membrane, it is possible to obtain information on the frequency of fusion events and the mode of vesicle fusion.
The main advantage of this technique over the existing methods, like for example electron microscopy and electrophysiology, is that it provides both spatial and temporal resolution required to detect vesicle dynamics. Indeed, highly contrasted images obtained by this technique allow to detect signals from single vesicles.